Continuing with the topic of pollen development, now we will talk about polyspory. Poly is many and spory refers to spores. So whenever there is occurrence of more than 4 spores in a tetrad, that is called as polyspory. We were just discussing about microsporogenesis, where a spore mother cell undergoes meiotic division, meiosis, to give rise to 4 spores. So a normal tetrad has 4 spores, but if any of the cells of this tetrad, it undergoes additional mitotic division, then the number of spores increases in a tetrad. So it doesn't remain a tetrad. The number of spores can be 5 or 6 or whatever. Hyphene is one example which you can see here. You can see that there are actually 5 spores which are present here. Now any of these spores can undergo another mitotic division and it can further lead to increase in the number of spores. The microspores in each tetrad, we just discussed that they are separated by deposition of callose. Once the callose is degraded, that is of course by the callase enzyme, these microspores, they are now released in the anther locule and they are present here in a partially dehydrated stage. Upon release, the microspore is now called as a pollen grain. So we call it a pollen grain only once it has been released from the tetrad. Once it is in the anther locule, it increases rapidly in volume due to absorption of nutrients and water. You would recall that we talked about two different types of tapetum, that is the amoeboid and the secretory tapetum. So the amoeboid tapetum, the remnants of the tapetum are already present in the anther locule, and if it is a secretory tapetum, then the secretions from the tapetum are there in the anther locule which nourish the pollen grains from here on, and of course there is water also which is there. So now in the second phase, this pollen grain which has enlarged because of the absorption of nutrients and water, this is going to become vacuolated, and the nucleus which is initially present in the center, because of the presence of a large vacuole, this nucleus moves to one side in a peripheral position. Most of the cell or most of the pollen grain is now occupied by vacuole or the vacuoles. The nucleus of the pollen grain will now undergo a mitotic division. So the first mitotic division in pollen grain, this is asymmetric, which will lead to the formation of a smaller germ cell which is also referred to as the GC, and a larger vegetative cell which we call as the VC. Most of the cell organelles, that is the plastids, the mitochondria, they remain in the vegetative cell because vegetative cell is the larger component of the pollen grain. The smaller germ cell is in fact, it is engulfed by the cytoplasm of the vegetative cell only. So generative cell can be referred to as a cell within a cell because it is a small cell which is present between a larger vegetative cell. The pollen grain, which now has a vegetative cell and a generative cell, can be called as a bicellular structure, or we can refer to as a bicellular pollen. Now once the first mitotic division is over, second mitotic division can take place in the germ cell, or it is the germinative cell, which leads to the formation of two sperm cells. The generative cell gives rise to two sperm cells or the male gametes. This process of gamete or sperm formation is called as the microgametogenesis. Now this is commonly called as microgametogenesis because there is formation of two male gametes. Here you can see that in A there is a tetrad of microspores, and with the dissolution of the callose wall, these are liberated from the tetrad and each microspore. So from the figure B onwards, this is one microspore. This is going to uptake the locular fluid and the nutrients, and it enlarges in size. There is formation of small vacuoles which you can see in B. Eventually these vacuoles, they coalesce or they come together to form a larger vacuole as is seen in C. Now because of the presence of a large vacuole, the nucleus is pushed to one side of the pollen grain. Now this nucleus will now undergo a mitotic division. The spindle which is formed is asymmetric, which has a broad side which is towards the periphery of this pollen grain. And then with the mitotic division, you can see that there is one nucleus which is now towards the center, and that portion of the cell is the vegetative cell, and there is a smaller one towards the periphery which is attached to the wall of the pollen grain, that is the generative cell. Eventually this generative cell will get pinched off and it comes to lie within the vegetative cell. So that's why we call it as cell within the cell. The vegetative cell is the larger cell, and this cell has no role to play in the reproduction. It is the generative cell which is responsible for reproduction. Vegetative cell always has a single nucleus. Now, at the time of the anthodehiscence or at the time of liberation of pollen grains, generative cell can be as such as one cell, or it can further divide and it may have two nuclei which are present. Vegetative cell nucleus does not undergo further divisions, whereas the generative cell nucleus always undergoes mitosis. The nucleus which is present in the vegetative cell is much bigger than the generative cell nucleus, and the nucleus of the vegetative cell is convoluted. In generative cell, the nucleus, if single, or nuclei, they are small. The DNA content increases in the generative cell. There is hardly any content of DNA which is present in the vegetative cell, and it remains the same. The vegetative cell is rich in plastids and mitochondria. Plastids and mitochondria are negligible or absent in the generative cell. Vegetative cell is instrumental or responsible for the formation of a pollen tube, in which eventually the vegetative cell nucleus and the two sperms are going to travel. Generative cell produces two sperm cells which are going to participate in fertilization. As you would recall that there are two sperm cells which are involved in double fertilization. One fuses with the egg cell and the other it fuses with the primary endosperm. It forms the primary endosperm nucleus by fusing with the two polar nuclei. There is lower amount of DNA and high content of protein in the vegetative cell, and there is high amount of DNA in the generative cell. Generative cell has high amount of DNA and it lacks RNA, whereas the vegetative cell is rich in RNA content. Vegetative cell is metabolically active and it accumulates starch grains. On the other hand, the generative cell is metabolically inactive, or it remains dormant except at the time of mitosis, and there is no accumulation of starch in the generative cell. Now, if the pollen are shed at the stage where there is a vegetative nucleus and a generative nucleus, then it is called as the two-celled pollen. And if it is shed at the three cell stage, that is when the generative cell has also divided, then it is called as the three-celled pollen, because the three-celled pollen will have one vegetative nucleus and it will have two sperm cell nuclei which are present. Two-celled pollen have a prolonged viability, whereas three-celled pollen have a very short viability. Two-celled pollen are easy to store, whereas three-celled pollen are difficult to store. Two-celled pollen are easy to germinate in vitro and under laboratory conditions, whereas three-celled pollen are difficult to germinate. Respiratory rate of two-celled pollen is low and that of three-celled pollen is very, very high, because the three-celled pollen are physiologically and metabolically more active than the two-celled pollen. Two-celled pollen exhibit gametophytic incompatibility. Three-celled pollen exhibit sporophytic incompatibility. Two-celled pollen occur in majority of the angiosperms and they occur in nearly 70% of the angiospermic families. Three-celled pollen are not very common. They are seen in Asteraceae, Lamiaceae, whereas two-celled pollen are seen in Orchidaceae, Nymphaeaceae, etc. Now, once the anther dehisces, the pollen are liberated and then of course they are available for pollination and eventually for fertilization. Most pollen, they are shed as single units and that is what we call as monads. But there are pollen which are shed as compound pollen grains, that is as units. They can be dyads, tetrads, pseudomonads, maculae, pollinia, pollinarium or polyads. Now, a single pollen grain which you see here is called as a monad. When the pollen grains, two are joined together and they are shed as a unit of two, we call it as dyad. Tetrad, of course, you know, will comprise of four pollen coming together, and then there are units of pollen where there are more than four pollen grains which are present. These can be polyads, maculae, or they can be pollinia, and we will be taking these up one by one. Monad, as I said, is a unit consisting of a single pollen grain. It is the most ancient type of dispersal unit found in hibiscus, radiscantia, vinca, etc. Dyad, as the name indicates, it's a unit of two pollen grains. Now, how do these two pollen grains they come together? These are fused either by the tectal layers or they are joined by some kind of mucilage or any other thing which brings them together. And basically dyad is a unit of paired pollen grains. Now, though the pollen grains are paired, but the pollen tube emerges from only one of the pollen out of the two pollen grains, and this is a very common feature of the family Podostomaceae. Here you can see that this is a scanning electron micrograph of some of the pollen dispersal units. B indicates a dyad which is seen in a member of Podostomaceae. In C you see a tetrad and D is a polyad, that is an aggregation of pollen grains which is seen in Acacia. A unit of four pollen grains which do not separate after meiosis. This is seen in Orchidaceae, Epidendrum. This is a tetrad. Now, when a unit of more than four pollen grains is there and the pollen grains are more than four but they are in multiples of four, we call it as a polyad. So the dispersal unit is then referred to as a polyad, which is common in Kaliandra, right? It has a polyad of 8 monads. In Albizia, the polyad has 32 monads, and polyads are also common in Fabaceae family. Pseudomonad is actually a monad, but this is not a true monad. It develops from a normal tetrad. Tetrad means there are four pollen grains which are present after meiosis. Post meiosis, Out of these four, only one nucleus remains functional, and the other three, they slowly degenerate, leaving only one mature functional pollen grain, which appears as a monad. Though it looks like a monad, it is homologous to a tetrad because there are four pollen grains, though the three have now started degenerating, and this is observed in Cyperaceae. Now this picture is showing the development of a pseudomonad. You can see that there is a tetrad which is formed, in which one of the grains it looks healthy, whereas the other three are degenerating. And in A, you can see that there is an arrow which is pointing to the degenerating microspores. So in B, this is a scanning electron micrograph of a pseudomonad in carex. The darkly staining region is the functional monad, and the white portion which you see here, these are the three microspores or the three pollen grains which are degenerating. So this is a pseudomonad. Then an aggregation of irregular number of pollen grains which are aggregated because of fusion, this is called as a macula. Not all the pollen grains which are present in an anther they fuse together, and these are mostly held together by viscous materials like pollen kit or tryphene. So I would like to mention that when we talk about pollen kit or tryphene, these are the pollen wall structures which we will be talking up later in this talk. Maculae are seen in Orchidaceae, and also there is presence of pollinia in Orchidaceae. Pollinium is a coherent mass of pollen which is compact and consists of many maculae which are united together, and these are united together and attached to a slender elastic extension. This is called as a cordicle. Now here you can see maculae in Pollinarium. Uh, the first one is a single macula in Orchidaceae, and the second shows two maculae with aggregated pollen grains. A collective term for all pollinia in an anther is the pollinarium. So all pollinia, they stick together by means of secretions or tissues. These could be visidium, cortical or stipe. It being sticky can get attached to insects during pollination. It is seen in Spiranthes spiralis. This is another picture of the pollinarium. You can see the cortical also over here. Now, another important feature of pollen development is the formation of male germ unit. So what exactly is a male germ unit? Male germ unit is an association of two sperms with a vegetative nucleus, and these two sperms are connected to the vegetative nucleus through a cytoplasmic extension. The existence of male germ unit is universal. It is commonly seen in Plumbago, Beta, Brassica. In two cell systems, the unit is seen in the pollen tube, as is seen in Gossypium and Petunia. Poaceae is one family which has no male germ unit which is present. Now the two sperms which are present or which are a part of this male germ unit, they can be similar in morphology or they can be different. When they are similar, we call them as isomorphic, and when they are different, we call them as dimorphic. Now these two sperms are connected with each other by common transverse wall. Only one sperm is associated with the vegetative nucleus through a cytoplasmic extension, and this sperm which is connected with the vegetative nucleus is called as the sperm 1, or it is also called as the SVN. The length of this extension is variable in different species, and it also is different at the different stages of the development. The other sperm which is not associated with the vegetative nucleus is referred to as the SUA. UA is unassociated, and this is also called as the sperm 2. The association between the two sperms and the vegetative nucleus is transitory and short. The reproductive unit is transported within the pollen tube and it is delivered to the embryo sac. Now this association of the two sperms and the vegetative nucleus is mainly formed to transfer and discharge these two sperms together into the synergid in order to prevent heterofertilization. You would want to know what heterofertilization is. Heterofertilization is when fertilization takes place by two different sperms from two different pollen grains. So if they remain together as a unit, then the chances of heterofertilization are less. The concept of this male germ unit was given by Russell and Kaas in 1981 in Plumbago zeylanica. The term male germ unit was coined by Dumas in 1985. You can see here brightly fluorescing two male gametes and a dull vegetative nucleus. Now this is a computer assisted picture of male germ unit in Plumbago, the first one you see here. On the left hand side you can see that there are two sperm cells which are present. The larger one has a cytoplasmic extension and then there is a smaller one, and on the right hand you can see how they are associated with the vegetative nucleus also. Lower down there is transmission electron microscope of male germ unit in Brassica, and on the right hand side is a transmission electron microscope of male germ unit in Nicotiana. Like I just mentioned that sperms can have a similar morphology, they can have the same shape, size, etc. So they are isomorphic sperms. If they are different, that is, then we say that there is dimorphism as is seen in Plumbago. So in Plumbago, this dimorphism occurs on the basis of size, shape, number, and also because of the occurrence of cell organelles in each of the two sperms. Sperm 1, SVN, or the larger sperm has a larger surface area, it has more volume, it has no or a very few plastids, but it has 311 mitochondria which are present, and this is connected to the vegetative nucleus, and this sperm is the one which fuses with the polar nuclei. The smaller sperm or SUA, which is not connected to the vegetative nucleus, it has up to 46 plastids and 52 mitochondria. This is the sperm which fuses with the egg cell. The two sperms, they remain connected through common transverse wall containing plasmodesmata. So because the two sperms are different and these two sperms they fuse with different entities, the male germ unit is a polarized functional unit. Now what is the significance of this male germ unit? As I said, that it is a universal feature of flowering plants. It directs the movement of sperms in the growing pollen tube. It also has to some extent a role in the germination of pollen and also in the growth of the pollen tube. Male germ unit may dictate the ordered arrival of each sperm cell to the embryo sac, so it ensures preferential fusion with the egg and the polar nuclei. So this sperm to sperm linkage may have a role in reducing the incidence of heterofertilization. So if the incidence of heterofertilization is taken care of, this results in embryo and endosperm becoming genetically different if the sperms are coming from two different pollen grains. So if there is no heterofertilization, the embryo and the endosperm, they are fertilized or by the sperms from the same pollen grain, and they are not genetically different. The association serves to deliver the two sperm cells simultaneously to the embryo sac, and it ensures a coordinated development of embryo and endosperm, facilitating crop improvement by genetic engineering of the sperm cell involved in syngamy and further utilizing it for in vitro fertilization. Now talking about the morphology of the pollen, this picture depicts a cross section of pollen which shows the inner wall layer, that is the intine, and the outer sculptured layer which is called as the exine. This is in ambrosia. Pollen grains can have different shapes and sizes. They can be rounded, they can be spherical, they can be tetragonal, they can be rectangular, they can be oval, so on and so forth. They can have different ornamentation. You can see that some have reticulations on the surface, some have depressions, and some are echinate on the surface. If we have to talk about the polarity in pollen grains, polarity can only be understood till the time the pollen are associated in a tetrad. Once the tetrad, they are liberated from the tetrad, we cannot understand the polarity of the pollen grains. So before we divulge into the details of the polarity in pollen grains, I would like to give a quick recap of the male germ unit, which is very essential for the fertilization in angiosperms. Two sperms, they travel together along with the vegetative nucleus in the pollen tube so that both the egg cell as well as the two polar nuclei, they get fertilized by the sperms from the same pollen grain, and there are no incidences of heterofertilization. So that is the importance of male germ unit, and we have also discussed the process of microsporogenesis and microgametogenesis in this class. In the next class, we will take up in detail the polarity of the pollen grains and the wall structure of the pollen. Thank you very much for your patient hearing and hope you have a nice day. Thank you very much.